Thanks, Roy. Argos is a public company. Okay, so Argos is built around our Arcelis technology platform, which allows us to induce a patient and tumor-specific memory T cell response, which others have shown that this specific phenotype of memory T cells correlates with good clinical outcome in multiple tumor types. And in fact, in our hands, the inducement of these cells, the absolute number, correlates with survival, PFS, and best tumor response. So we know exactly how our therapy works, and when it works, we see the clinical benefit, which we think is unique in the cancer immunotherapy space. We've taken that technology and moved into now a phase 3 trial in first-line kidney cancer. We're combining with standard of care. AGS-003 is our product, and I'll show you where we are on that trial in a second. We are also moving AGS-003 into other settings in kidney cancer beyond the metastatic clear cell population. We're going into non-clear cell as well as earlier stage kidney cancer, and also going into bladder cancer and non-small cell lung cancer. All these trials will start this year. We've also used the platform to develop a product for HIV, where we're in a phase 2 study where we're trying the kick and kill strategy, combining with an HDAC inhibitor to try to flush the virus out and then eliminate it. And that trial should be moving into stage 2, where we introduce the HDAC shortly. We have developed an automated centralized manufacturing process. We think we're way ahead of everybody else with autologous therapy in that, and that'll be in place prior to BLA filing and allow us to launch in a cost-effective, scalable manner. So the phase 3 trial is going to finish enrollment this summer. We're at, I think, around 350 patients today. We need 450 to finish. We have over 75 patients in screening right now, so we need to screen another 100 or so to get there. So maybe another couple months of screening, and then in randomization enrollment, and we'll be done. We have our first interim at 25% of events. We have an IDMC meeting scheduled during ASCO, where we expect to have at least 25% of events at that point in time. And then we have subsequent interims at 50% of events, 75%. And if we go to 100% of events, that'll happen in the second half of 2016. And as I mentioned, we'll also be moving AGS-003 into other indications in kidney cancer, as well as bladder and non-small cell lung this year as well. So I just want to skip to this slide. So the product or platform is made up of two things that we get from the patients. The first is a tumor sample. It can be a biopsy. In this case, we're talking about newly diagnosed kidney cancer patients who need surgery, so we have more than enough tissue, but we can use just a few milligrams because we're taking just the messenger RNA, but we're taking all of it, and then we're using PCR to amplify it, so we get more than enough to treat the patient for life. And this gives us a perfectly matched antigen base to the patient's disease. And the second thing we take is a leukapheresis from the patient, which we then isolate monocytes, and we have a proprietary process where we end up with a dendritic cell, and you see the CD40 ligand that we supplement with, and that overcomes the immune suppression, and I think we might talk about that a little bit with Roy. But the result is you get this highly optimized dendritic cell population that's expressing all of the tumor antigens, most importantly the mutated antigens, which is really where we differentiate ourselves from everybody, and because we think it's the mutated antigens that are going to drive an immune response, specifically that memory T cell response that is tumor specific, and that will result in clinical benefit. Our potency marker is IL-12, and it is, as far as we're aware of, we're the only ones who can generate this specific memory T cell response, and the fact that it correlates in our phase 2 with survival, PFS, and best tumor response makes us confident that when it does work, it results in clinical benefit. So that's my introduction, and I think now we'll go into some Q&A. Thanks.
Hello? Okay, great. Thanks, Jeff. So yeah, I think they did it. So the first question, let's go into the differentiation of Arcelis versus, and I think you have a slide in here, correct?
Yeah.
Let's go to that slide.
I do. I think it's a...
So this is a corporate slide deck, and it's on the web.
And it is on our website, right. Yeah. Yeah, so let's use this slide to talk about the differentiation of our approach versus others. And as I said, first and foremost, by taking the RNA directly from the patient's tumor, we're specifically targeting the patient's disease. We're expressing all of those antigens. And I think most importantly, as I said, it's the mutated antigens. And this obviously is what differentiates us from Provenge and Oncotherion and all of those approaches that came before, where you're targeting non-mutated antigens that happen to be overexpressed, but they're not mutated. And T cells are eliminated when you're a child. Your immune system is taught not to respond to non-mutated antigens. T cells that respond to non-mutated antigens are eliminated from your immune repertoire. So that's why you've seen an inability for real clinical benefit from those approaches, as well as an inability to show a correlation between immune response to a non-mutated antigen and any clinical benefit. Second thing is you need to overcome immune suppression. Obviously, there's different ways to do that, and what we found is by optimizing the dendritic cells, and specifically with the CD40 ligand that we include, we're able to get a potent immune response. We see patients who at baseline can't respond even to flu antigen, not to mention other cancer antigens, but who now can respond to normal antigens like flu with an adaptive immune response. But of course, most importantly, we get the memory T cell response to the tumor itself, which is what we're looking for. But we actually have this additional benefit of overcoming general immune suppression that's there at baseline in these kidney cancer patients as well. Obviously, we believe the future of cancer therapy is combination, and that's where we have a huge advantage because we have virtually no toxicity. From all the studies we've done in cancer and in HIV, we've never had a serious adverse event associated with our product. So this allows combination. Just what we're doing in kidney cancer, combining with standard of care, targeted therapy, Sutent, and the others, all very toxic. None of them can be combined with each other. That's where we step in. So we offer oncologists and patients the benefit of taking AGS-03, starting with Sutent, switching to whatever they want at any time, and keep going. And then the last point is the broad applicability. What we do in kidney, that's what we're doing in bladder, that's what we're doing in non-small cell lung. The automation, the manufacturing, identical. The people who work at Argos don't even know what tumor type they're working with when they make a product. It's the exact same product.
Okay, great. Now, so you skipped it, but let's focus on the bottom right corner here for a second. Maybe how do you get this product up and scale it profitably versus, say, Provenge?
Yeah. So obviously the big question that people have, yeah, looks like it works. Yeah, this is pretty cool, but how are you going to make money? So a couple things that are important to making money with autologous therapy. I think first and foremost, it's logistics, and second is cost of goods. And so what we've designed is a process that addresses both. As you can see on the slide, our only limitation in terms of logistics is we need to receive the leukapheresis in 4 days or less. Our phase 3 trial is being done as far away as Israel, all from North Carolina, and we haven't had any problem receiving material and shipping material. From one batch of cells and a tumor sample, we create on average 3 years of therapy for patients. Unfortunately, that's enough for most patients for life with metastatic kidney cancer. But for patients who've lived long, and we've had many who have, we just bring them in for a second pheresis in the 3rd year. Sometimes we get more than 3 years of doses, of course, and then we make the product again and we keep going. The final product is frozen. We ship in a temperature-controlled shipper that keeps it at target temperature for almost 2 weeks. Obviously, that's more than enough. And so we have all the logistics, and then when you take our automated process that we've developed, you then get to a cost of goods and a scalability that allows you to address not just kidney cancer in a profitable fashion, but breast cancer, lung cancer, major tumor types as well.
Okay, why don't you give us an update on enrollment in the phase 3? I think one was just made at ASCO GU.
Yeah.
And what we can expect maybe from this 25% interim.
Sure. So I just want to point out one thing from the phase 2 when we talk about enrollment, or maybe two things. So this is our comparison of our single agent phase 2 and our combo. Obviously the combo is the critical one on the far right because that's what we're doing in phase 3. But I want to point out that our median OS in the intermediate risk patient groups, which is on the sub-bullet on the far left, which was just over half the patients, was almost 5 years. And that compares to an expectation of 20 months for patients treated with targeted therapy, so about a tripling. And to Roy's question, we have designed the phase 3 to enroll primarily intermediate risk patients. We think those have the best chance of responding successfully to our therapy. The other point I'll make is on this slide, this is all the patients in the phase 2. On the top left, you can see the poor risk patients in the dark bars, 5 of whom received 4 or fewer doses of AGS-03 because of the design of the study. If a patient progressed, and we were scanning patients frequently because of Sutent, they came off Sutent and they came off the study. In phase 3, because we're doing a survival study and because we've shown we can combine with the other agents, patients can now continue dosing if they switch off Sutent for any reason, progression or toxicity. So in the phase 3, we're now exactly where we want to be, which is and the last point is we've eliminated the poorest of the poor risk patients, those with 5 or 6 risk factors. Those patients live for only a couple months and are not good candidates for this trial. So with the trial now at around 350 patients randomized, we have 73% of those patients intermediate risk patients, 27% poor. Almost 50% of the patients have 2 risk factors, meaning on targeted therapy alone, those patients live for only 12 to 14 months. That's our sweet spot. Then about 25% are 1 risk factor, or 20%, 23% are 3 risk factor, and then the remainder are 4 risk factors. So we have exactly the patient population we think will benefit best. If we do half as good as we did in phase 2, we still have an incredibly successful trial because our primary endpoint is about a 6-month improvement based on that hazard ratio. So to Roy's direct question, as I said, we'll finish enrollment early summer. The interim will be at ASCO. 25% interim obviously is very early. As I said, we're probably going to be closer to 30% of events. That's still pretty early. I think we expect to see the IDMC meet and say everything looks good, trial continues as is. When we get to the 50% interim towards the end of the year, assuming we get a positive read there, now it's quite interesting because you know that the experimental arm is doing at least, pretty, I won't say significantly, but doing better than the control arm for sure at that point. And then we're 6 to 9 months from final data. So that's the timeline for the trial and where we're at with enrollment.
Okay, maybe really quickly, what other combos besides Sutent are you considering? Anything we can look for in the near future? Yeah,
so not surprisingly, we're working on a combination with a anti-PD-1 or anti-PD-L1 in kidney and bladder and in non-small cell lung. So hopefully one of the trials that we launch this year will be in combination with a checkpoint inhibitor in bladder. And then we're going to also be combining with standards of care, whether it's chemo, platinum-based chemo. We see synergy actually with platinum-based chemo in our approach. So that looks good for bladder and non-small cell lung we're excited about. So as I said, the advantage is we can combine with pretty much anything, but we do see a real benefit in combining with the checkpoint inhibitors, because what we do is induce new memory T cells that are not existing at the time the patient's treated. What those do is unleash pre-existing T cells, which we don't impact. So it makes sense.
Okay, great. Thank you, Jeff.
Thanks, everybody. Thank you.